Hi, my name is Sulov and I'm from Bangladesh. So right now I am doing my PhD in France. The city is Toulouse. It's almost at the south border of France, so right next to Barcelona. And yeah, the I'm doing my PhD here in basically in developmental neuroscience. So studying the role of nutrition and the developing brain. So how nutrition can affect in the build up of a brain.
Okay, that's cool. So before joining your program where you have been, before starting your PhD.
So I completed my bachelor in Bangladesh in Maulana Bashani Science and Technology University. Then I moved to Germany for my master's program, and the program was on translational neuroscience. So there I had exposed to neurodevelopmental studies and research. And yeah, it was a good experience. And yeah, right now I am here in the same field. You can make an organoid from any organism, so it's not very specific to human. But as we want to find out the pathology related to human and developmental regulation related to human to better understand how it is going actually in human, and then we can use this knowledge to overcome some pathological condition or disease. So I am using human cerebral organoids. So it's basically something that is very similar to developing brain of human.
So if I understand you, so normally what we hear in kind of mainstream news, like artificial brain, artificial kidney, liver. So is this organoid the same thing like we have like artificial brain?
Uh, yes, it's artificial, of course, because the source of this organoid is basically an adult cell that we use iPS. So we uh, do a induced pluripotency in that cells. So they become more, they get the features of embryonic features, and then we can differentiate to any kind of organoids or any kind of tissue, any kind of cells. So yeah, it's kind of artificial because it's actually not 100% mimicking the same tissue in living organism, but it's the closest model right now we have in human if we want to study some living cells in human, because either you are investigating human cells from from postmortem samples, so that's not uh, also a living cells. So it's really hard and a matter of ethical aspect to investigate human samples. And I think on this point, Organoid is doing a great job. So giving some real information what happens in human background, and like there are some specific genes that is expressed only in human that we cannot address that question in mouse or something else. That might be better if we go forward with organoid. It's an adult cell, so we first we have to convert it to iPS cells,
Mhm.
and then it's like more like a stem cells embryon. So they are almost homogeneous, and after that we have to give them some direction to go into uh, like neural path. Actually, there are four different mediums that we have to put them, like depending on the cell state. So first, like an iPSC medium for culturing the iPS cells. to make them a cluster. Then we make them an embryonic body, so they are making a cluster. So that's in another medium, and then we put them into the neural fate specification medium. So they are becoming uh, going forward to make the brain organoid. And after that, we just have to make them mature. So there is a medium for maturation. Like this maturation medium will promote the differentiation of neurons.
So, do you have anything like what is your project? You can share your um, maybe slides or what study you was talking about.
Mhm. Iodine, this kind of macronutrients, micronutrients are essential for the development of the brain. And that's where our research lab is focused, only specific to the folate, which is also known as vitamin B9. So how this uh, folate can help to develop the brain. Because so far we know that when there is a deficiency of folate, there could be a phenotype that you could have a smaller brain or a large brain. So it's not normal when there is a deficiency. And this is a case of severe deficiency of folate. But in our lab, we want to investigate now when there is a deficiency of a little bit. Like let's say when someone is pregnant and not taking folate for a couple of days or for a week only, could it affect the developing brain? That's our main research question. And for
So
that,
it's like a
yeah,
malnutrition of vitamin B. So
yes,
if pregnant mother don't take enough folate, what will it affect his baby or not? But in
yes.
general, when you totally don't give the folate, that have a serious effect. So is there any relative disease or anything like what kind of effect like growing is just abnormal
Uh,
brain development or anything like in later
yes.
life is that disease?
So, like in the case of a smaller brain, there is a possibility of that the child could mm, not born alive. So maybe they will be dead at the time of birth. And in the case of like also the overproduced large brain, it's also that this child will be affected, and like the expectancy is very small, like couple of years only, maybe maximum.
Okay,
So it's a huge effect.
that's good. Okay. Mm.
And the earliest form this deficiency can happen is like in the case of human, it's like 5 weeks uh, of the pregnancy. So let's say uh, before 1 month. And in most cases, the women might not be knowing that she is pregnant. So she is not careful enough to take the folate necessary for the embryo. And that's where the deficiency is much more prominent, especially a country like a developing country like Bangladesh. It's we we can see this type of phenotype uh, very often.
So this folate actually that you said more severe in early stage of pregnancy
Yes,
rather than the later stage. So
yeah,
even sometimes they don't know.
yeah. As as I said, the brain structure starts to form around 1 month of pregnancy. So and then the brain develops until birth. So it's a huge timeline to maintain this. diet necessary needed for the embryo. So we have to be very careful, and we have to be very specific that know when folate is essential and how much do we need, because it's also interesting that uh, a lot of folate can also be detrimental, so it's not also good. So we have to balance our diet properly to maintain the level.
Okay. Hmm.
And in the next slide, what I am to show is our research model, how we are addressing our research question. So on the left, it's the pregnant mother. So she can take folate in the form of green vegetables mostly, and this folate can pass through blood and placenta and then help in the build of developing brain in the embryos. And this is a in vivo situation. And on the other hand, we have another model, the cerebral organoids, that is in vitro. And what you can see here, like this on structure is like one hemisphere in the normal brain. So this is a mouse
Okay.
brain, of course, but if you consider it as a brain, this structure is one hemisphere.
Okay.
So in one organoid, like here, you can have several of them. So it's like in one organoid, you have multiple brains. And like in the middle, here are the dead cells. So there are some cells that are not getting enough nutrition, or there might be in a stress condition in the medium, because it's a in vitro situation. So there are some dead cells, and they accumulate at the middle of the organoids. But apart from that, this structure is very similar to the developing brain, even in human. So also in human, we see this structure and this level of organization, and it's quite same. So that's why we use this cerebral organoids model to mimic the human development.
Okay. So mainly you use two model. One is mice model that is in vivo,
Yeah,
what happening there, and at the same times you taking those research, those findings in the organoid in vitro cultures, and as you said, it's kind of many microbrain there,
yeah,
and this microbrain is kind of mimicking the actual human brain, and you can see whatever you can see the effect of folate or vitamin B9, right?
yeah,
Okay.
yeah. In the case of like in vivo or mouse situation, like you can model the research question on your demand. So like you can create some deficiency in the nutrition, or you can create some deficiency in the gene, like we are doing right now. Like you can target some specific enzymes in the metabolism, so the pathway is not moving forward. It's also mimicking same as the nutritional
So you
deficiency.
mean pathways that are related to making vitamin B9? So some you block some of these
Yeah,
pathways.
yes. It's interesting that we cannot produce vitamin B9 in our body, so we have to take them as a form of food. And after we take them as a form of food, there are some enzymes that convert this folate form in the diet to different metabolites, and that metabolites are necessary for uh, essential cell functions like DNA synthesis or like methylation reaction, this kind of functions.
Cool. So how you in case of organoids in vitro, how you give this folate? I mean, just um,
Yeah. So if we want to model at the nutritional level, we can make some diets like with extra folate or with no folate, and then give it to the pregnant mother, and they will eat very similarly. So same amount of food they will eat, but after a few days, uh, yeah, there will be a deficiency in the pregnant mouse, and that deficiency is affecting the mouse pups. So that is our
that's
plan.
cool. So you give them different concentration of folate and see
Yeah.
the effect of it. And as you trying to find out maybe the optimal concentration, so and
Yeah.
most importantly, how you evaluate them, like what kind of assays you do, or what kind of test you do that it's happening, especially in vitro.
So first we have to check if the mice is eating the new diet properly. So we check by measuring the diet consumption,
Mhm.
and if it is normal, then we can proceed to the next level. Okay, now they are taking the same amounts of foods. So if there is an effect in the pups, so it should be because of the nutritional deficiency, not because they are eating a different diet or eating a less of this new diet. It's not the case. So then we take out the mouse embryos at different points and analyze their brains with immunofluorescence. So how sometimes we can see the effects on the structure, like in the morphology. If the brain is smaller, very much smaller, we can see comparing to the normal brain at that stage. But in the most cases, the effects is not that much prominent. So the effect could be very specific to some cell types. So even if you have same size of the brain, you could have a different brain because there are different cell types produced than the normal all, and their connectivity is totally different. So that is on our main research goal to identify how a nutrition could produce a specific type of neurons, and if we consider this on a big brain, how it is happening like in human. So
So
that is the
it's like
translational
sometimes
part. Yeah.
you check with immunostaining, immunofluorescence staining of the brain's slice, and sometimes you can even see those effect on those immune staining, but sometimes you couldn't because apparently their size and structure is same, but
Yes.
their molecular features or markers is different. Maybe they have different kind of cell type or
Yeah.
as it growing brain. So you want to characterize those features. So is there anybody have done this kind of work before, like similar work, the effect of vitamin B1 in the human brain?
Um, no, not especially in the brain cell composition. It hasn't been done yet, so we don't know how whether a specific nutrition can produce a specific neurons. Because let's say if the mouse or if in human, like if we are producing with folate a little bit of lead bond neurons, and the lead bond neurons functions like that, so they connect one hemisphere to the other hemisphere mostly, rather than the early bond neurons. So they are the early bond neurons connect from one hemisphere to the spinal cord. So they are mostly regulating the motor behavior and things like that, rather than the um, lead-borne neurons that they are connecting to the interhemisphere and making decisions. So there are
So
huge
you mean
difference.
you mean that mostly it affect the neuronal connectivity during
Yes,
developmental phase, because as it grows, like billions of neurons have different kind of connection and plasticity. So mainly it affect those connections or the cell populations among the brain. You also have recently present a poster in an international conference. So can you tell us a little bit more on this, like where is the conference and
yeah.
what
So
it's
this was the
about?
poster,
Mhm.
and I presented it in a Keystone Symposia conference. So it was this week, so last week. So um, yeah, it was based. The conference was based on metabolism, and as we are addressing the question in metabolism, so it was good for me to expose into some classical metabolic studies and research how they do it. So
More
yeah, it was
importantly, what they think about your research, right?
yeah, yeah, that's true. We want to know the feedback uh, from the people who are specialized in metabolism. So how they are feeling about this project, and if there's something that we can improve.
Okay.
And basically in the poster, I presented the results that we have got so far. So not everything, but I presented like results from our heterozygote mouse. So it is heterozygote for on enzyme that is essential for folate metabolism. And so it's a heterozygote mice. So the enzyme is active around at 50%. So 50% activity is reduced. So it means there is 50% deficiency of folate metabolism. And in the mice, we have already characterized the phenotype. So we are seeing that the progenitors production is affected because this enzyme is very specific to the progenitors, neural progenitors. And as the progenitors is affected, this progenitors means the stem cells. As the stem cell is affected, the neurons produced from these stem cells is also changed. So we are seeing that there are less neurons connecting that should connect to the spinal cord, and there are more neurons that connect to the other hemisphere. So there is a huge difference in the brain uh, if you consider the cell types. But as I said, in general morphology, uh, there was no difference. So the brain was same in size compared to normal brain, and the thickness was same, but only the cell types in the brains was different. So this was very interesting. And yeah, that was the results from mouse. And so far we have done uh, not much experiments on cerebral organoids. We are just characterizing the things we are seeing in mouse. And as we know that human development is much longer regulated, so we are characterizing uh, how long this folate metabolism is necessary, or how long it is active in the cerebral organoids. So we are characterizing this right now. But uh, the results are showing promising also in cerebral organoids. So hopefully we will be able to characterize it properly and completely soon.
So that means um, you want to say that we are talking about before that which stage probably during the development this
Yeah.
folate have a more severe effect. Like
Yes.
for humans, that is a 10 months if you see even during the pregnancy. Within 10
Yes.
months, which weeks or which months it have a like um, robust impact in the development.
Yeah. True.
Okay, so and those features like the box uh, plot, it's just like a different kind of yeah, that looks good now. So in two in research question two, we see that they have different cell type, right? This different color.
Yeah.
Okay. Mhm.
So
But
that's
one
how
question.
basically is it's done in mouse embryos. So in mouse embryos, there is a neurogenesis period of 6 days. So all of the neurons are produced in within 6 days, and in human it takes 4 months. It's basically the uh, same neurons, but they are huge in numbers, and yeah, they are taking much more longer time to produce more numbers of the same types of neurons uh, in human.
Okay, that's also. I have a question on number three, like the generations of heterozygous mice. So you do it with CRISPR-Cas9 technique or Krylux
Yep.
technique?
Now we did it with CRISPR-Cas9. So we first created the like deleted one of the exons from the gene in the zygote, and then we put the zygote back to the mouse um, mothers. So there we get the pups that is heterozygous.
Okay. So like sometimes we see when we delete one allele or like make um, heterozygous. In that point, the other part, the other copy is become try to what is it called? Rescue it, try to overexpress. So did you see any kind of effect like is it trying to
Uh,
overexpress or is it like same like before?
uh, no, it's actually compensating somehow by like still like we are seeing that there are the expression is there
Hmm.
and the activity is also there. So what we are seeing in the heterozygous mouse that the activity is reduced in half. So the other allele is active. That means, and also in the Western blot we are seeing that there is less protein. So yeah, it makes sense that the other allele is only active and they're somehow trying to compensate.
So the number four is the immunofluorescence pictures are from the brain in like a day 12, day 14, and day 16 different phase in
Yeah,
comparing to wild type to these heterozygous.
yeah.
Yeah.
Basically, here we characterize the stem cells in the brain.
Mhm.
So whether there is any changes in the red or green population at a specific time point, because as you are seeing at the beginning, the red population is higher because they are the founder population, founder stem cells, and
Mhm.
these red cells will produce the green cells later. So if you see the green cells are now getting higher in numbers, and both of them can produce neurons directly.
So the red cells, you mean the early stem cell progenitor, and the green one is the late progenitor.
Yeah,
How do you mark them? I mean,
there are some transcription
antibodies.
factors.
Okay.
Yes, uh, transcription factors that is very specific to the cell types. So the PAC6 uh, is for the red. So yeah, we can identify with the by using antibodies again these markers, and also we know the localization, so we can characterize by it because you can see here the red cell should be at the bottom. They are connected to the ventricles. Ventricles where is the nutrition source. The blood, the ventricles contains the nutrition coming from blood. So these are the cells directly connected to the blood nutrition, these red cells. And these red cells will divide and produce the green cells that will be at the top of them. So they will form a defined brain regions. So we can identify them easily, how much they are, and yeah.
Okay, that's nice. Yeah, that you said like uh, in terms of structure, they look similar.
Yeah.
That is not.
And as you can see, this is day 12, day 4, and this is day 14 and day 16. So at day 12, the brain tissue is smaller. It's expanding little by little over time. So that's why you can see more bigger staining later stage. And we also characterize the neuronal cells. Basically, these cells are blue. These yellow and red cells are blue in the first staining because we didn't stain at in that same sections. So this is defined sections, but at the same stage, here we stain for defined neurons. But the idea is very same. So we have some transcription factors specific to specific neurons. So we just target them with antibodies.
Cool. Yeah, that's it just
And
looks so beautiful.
as you can see at day 16, the cortical structure is almost same. The thickness, so the size is same. This is normal brain and this is heterozygote brain.
So all is only just changing the populations a little bit. Like in the earlier, there is no significant difference, but in later progenitor, you see like more green cell, less red cell,
Yeah,
something like that. Their number is quite different.
yes.
Especially in the day 16, it looks more prominent
Yeah,
that their
because
cell populations.
at day 16, almost all the neurons are produced. So at that time, we can see the actual phenotype if there is something. Because at day 12 and day 14, it's very early stage of neuronal production. But at day 16, it's almost done.
Okay.
And as I have said, we are trying to model it in human cerebral organoids. So we are trying to model it right now with in using an inhibitor of DHFR of the enzyme we used in the heterozygote mice. So we are inhibiting that enzyme with a drug. This is actually a cancer drug, but specific to that enzyme. And when we treat them with this drug, we are seeing that there are some changes in the neural production. So we are seeing more neurons are produced compared to the normal cerebral organoids. So this we have to characterize which type of neurons are produced especially, and we also have to uh, characterize when we should treat the organoids with the drug. It also varies the result. So this will be the future work we are focusing on. And at the end, I just conclude with something what we are seeing as a phenotype and what could be the reason. So we are hypothesizing how the progenitors is affected and finally giving the phenotype.
Hmm. Yeah, that looks so important. I think that's a great work, especially so your task now to maybe the more characterize those cell populations and their functions probably.
Yeah, the right now we are addressing the question how uh, why this type of cells are affected because not every cells are affected similarly. So what's the reason behind this? So we are going through gene by gene now, right? So to see, yeah, whether a set of genes are affected due to the deficiency, and that's why maybe they are changing their cell fate.
So do you have any plan to run like single cell sequencing, RNA sequencing, just to find
Right,
out?
yeah, we are trying to do some chipset analysis. So
Hmm.
this is for identifying candidate genes if they are regulated differently. And also we are trying to do some ataxic. So this chipsec and ataxic are almost very same. Um, so we will just analyze whether there are some specific genes that are activated or inhibited in our mutant phenotype. So we can easily identify this gene and then study the mechanism very precisely.
Yeah, I like those. But that's really wonderful work. That's excellent. I mean, it's always hard to do a study on the brain sections, and because we don't have enough samples, and it's not like other tissues that you will take and do whatever you want to do. It's really tough to quantifying them.
Yeah, yeah, that's one of the issues that uh, you have to be uh, very precise and very you have to be very technical throughout the procedure because if one process is not done properly, you can see some variation. So yeah,
I mean,
that's
it's hard to reproduce the
yeah,
same data, right? Because there's lots of challenge, technical challenge, and if you can't reproduce it, it's not acceptable to the scientific community. So you have to be
yeah,
make it reproducible.
yeah. That's one of the important issue right now in the research field that to reproduce the data. But yeah, everyone is trying hard. That's where the protocols and following instructions, everything like these are helping. This can help someone to better reproduce something.
And I think I would say it's also because of these days that there's lots of big data. Like if you analyze, suppose chipseq or ataxic or say, single cell sequencing, even like this microscopy image, like immunizing image, like which one you are reporting because you got actually lots of images. Maybe you are just reporting two or three as a representative. So
Yeah,
how unbiased you are selecting those
yeah,
one? Are you just selecting that are have giving you something
true.
phenotypes and showing? But yeah, that's
Yeah,
the honesty is
that's
also important.
true.
Okay, thank you so much to sharing your science and the work with us.